Hello and welcome. I'm Pam Silver, and I'm a senior editor at M Systems. My group works broadly in the area of synthetic biology, where we seek to find living solutions for problems facing the world in both health and sustainability. We often find ourselves turning to microbes because of their exquisite ability to live in different environments, to sense and respond to their environment, and in many cases to be highly engineerable. For that reason, I'm thrilled to be here to open the fourth webinar in synthetic microbiology for M Systems. This is an exciting area for the journal, and we hope that this webinar will inspire you. We have three amazing speakers. The first is Wenying Xu, the second is James Carruthers, and the third is Carolyn Aho Franklin, who is an editor in this area at the journal. So I hope you enjoy the day.
Hi, it's my pleasure to introduce you to Dr. Wenying Xu. She is a professor of quantitative and evolutionary biology at the Department of Genetics, Evolution and Environment and Center for Life's Origins and Evolution at University College London. Wenying graduated from Pomona College in Southern California, double majoring in molecular biology and mathematics. She then went on to Caltech for her PhD, in which she studied how budding yeast complete the cell cycle. Afterwards, she performed postdoctoral research in a physics and biology lab at the Rockefeller University, where she became interested in microbial cooperation. She then started her lab at the Division of Basic Sciences of the Fred Hutchinson Cancer Center Research Center in Seattle, Washington. Her lab uses engineered yeast communities and in silico communities to understand how cellular cooperation can persist against cheaters, how community-level properties emerge from interacting individuals, and how to evolve communities with improved functionality. Without further ado, I will turn it over to Dr. Xu.
Hello, I'm Wenying Xu from University College London. Today, I would like to share with you lessons we have learned from a synthetic community. Synthetic communities can be used for biotechnological applications. But today, I want to focus on how we have used a synthetic community to understand questions from basic biology. The basic biology question we were interested in is the evolution of cooperation. I will also share with you the lessons we learned about the importance of quantitative thinking. Cooperation is ubiquitous in biology. From independent replicators, that is, bits and pieces of nucleic acids that are capable of self-replicating, to being strung together to form chromosomes where replication is synchronized. From unicellularity to multicellularity. From individuals to society. Each of these major transitions in evolution involves cooperation between lower level units to form a higher level collective. But the cooperation is threatened by cheaters. In a society, we have thieves. For multicellular organisms, we have cancers, cells that grow more than what they should. And for chromosomes, we have transposons, which replicate more than what they should. How might cooperation survive cheaters? I want to more rigorously define constraints of cooperation. So let's consider the net gain. When there's no cooperation, the baseline, the net gain is 0. A cooperator pays a cost C to generate a benefit B greater than C. So if this cooperator interacts with another cooperator, then the net gain would be B minus C. And because B is greater than C, B minus C is greater than 0. A cheater pays 0 cost and generates 0 benefit. If a cooperator interacts with a cheater, the cooperator's net gain is 0 minus C, or simply minus C. The net gain of the cheater is B minus 0, or B. We can see that B is greater than B minus C or minus C, and thus cheater frequency will increase over time until the community is consisting of purely of cheaters, and in which case we will return to the 0 baseline. we can immediately see that there are two constraints. First is the partner availability, whether this cooperator can encounter another cooperator. And the second is cheater invasion. Today, we will focus on the second constraint. How might cooperation have evolved to cope with cheaters? We use an engineered yeast cooperation and cheating community to address this question. So the community consists of two yeast strains. The first one cannot make lysine, but overproduce adenine. The adenine overproduction phenotype is caused by a mutation in the first enzyme of the adenine biosynthetic pathway. And as a result, this enzyme is no longer sensitive to end product feedback inhibition control. So basically, cells do not know that they already have enough adenine. They just keep making adenine. And you call this L-A+. The complementary strain cannot make adenine, but overproduce lysine. The overproduced metabolites are released into the medium. A side note that it's actually not adenine, but hypoxanthine that's released. But for simplicity, we'll just call it adenine. The hypoxanthine is a derivative of adenine. And we call the system Cosmo for cooperation that is synthetic and mutually obligatory. The two strains are non-mating and thus can be regarded as different species. We also label them with different fluorescent proteins so that we can distinguish them apart. Importantly, we can engineer cheaters. So instead of L-A+, we have L-. So those cells cannot make lysine. They make adenine, but just enough for itself. So they do not release any adenine. And the cheater has advantage over cooperators because we can mix cheaters with cooperators in excess lysine, and we see that cheaters grow faster than cooperators because the ratio increases over time. I argue that Cosmo is a model system for studying naturally occurring cooperation. This is because nutrient exchanges are common, for example, between legumes and the nitrogen-fixing rhizobia, between host and gut microbiota. If we generalize nutrient exchange to benefit exchange, we have many more examples. For example, the fig flower and the pollinator fig wasp exchange benefits. By capturing a feature that is common to all these systems in which a partner had to pay a cost to supply the other partner with a benefit. So the exchange of costly benefit. Because Cosmo captures this common feature, it will allow us to learn principles that are general to all of these. Over the past years, we have indeed learned general principles, and I will share two vignettes with you. The first one is that strong cooperation promotes species coexistence. So here's an experiment where we mix two strains at initially 1,000 to 1, 1 to 1, and 1 to 1,000. And then we see that over time, the strain composition approaches the steady state of 1. And this actually makes intuitive sense. Suppose the green population is much fewer than the red population. So what happens is that there'll be a lot of red benefits, but very little green benefit. And these cells have to compete for very little benefit, and this rare population will have a lot of benefits. So as a result, this will grow faster than the minor population will grow faster than the abundant population, and the ratio would be reset back to the steady state value. So stable ratio has also been observed in natural mutualisms, for example, in methane generating community and in anaerobic methane oxidizing community. The second general principle is that strong cooperation promotes species intermixing. So here we have both simulations and experiments. So in experiments, we just mix the two strains and allow them to grow in agar pad without adenine or lysine. So they're forced to cooperate. We see that the red and green cells form these intermixed patches. In contrast, if we supply rich medium to the agar pad so that these two strains compete, then we see beautiful columnar patterns. They form columns in both simulations and experiments. This result is also intuitive in the sense that if two populations are cooperating, they will try to grow toward each other. to have better access to the metabolites. And this will cause intermixing. But in competition, there's no such nutrient exchange. So there's no need to grow toward each other. So they form this boundaries, the straight boundaries. So this intermixing between strongly cooperating populations have also been seen in natural communities. In this example, disulfur vibrio, DV, produces hydrogen as a byproduct of its fermentation. This hydrogen is inhibitory to its growth, but this hydrogen is useful for methanococcus. So methanococcus helps DV to grow by taking away the inhibitory hydrogen, and DV helps the methanococcus to grow by providing the reducing power. And in fact, if we look at the DV and MM, the disulfur vibrio, the methanococcus, they form intermixed pattern. So I've shown you how we have used a synthetic community to learn principles that are general to other systems. Now I will show you an example of how we use the synthetic community to discover new mechanisms. So there are known mechanisms by which cooperators can be protected from cheaters. The first is partner choice. So suppose if I'm interacting with two individuals, and I can recognize this one's cooperator and that one's cheater, and I will only interact with cooperators. So this would stabilize cooperation. But our yeast community is engineered and does not have this partner choice mechanism. A second mechanism is a spatially structured environment. So this is experimental view from the top. We can see the red and green cooperators intermix to form these pods, and the cheaters are isolated. And this is intuitive as well, because the two cooperators will grow toward each other and form intermixed pods. But if a cooperator happens to land next to cheaters, the cheater does not provide anything. So the cooperators would not grow well, would die eventually, and the cheaters would eventually also die. So cheaters cannot grow very much. So this would protect cooperators against cheaters. But we can experimentally easily get rid of spatially structured environment by simply shaking our culture tubes. So third mechanism is privatization. So suppose I'm making some benefit for others. If I save a little bit for myself, this can already make this whole thing profitable. But note here that each strain needs the metabolites from the partner. So privatizing the self-made metabolites is of no use to the cell. So this is not relevant. So now we can ask what happens to Cosmo in an environment known to be unfavorable for cooperation. So the experiment is performed in the following fashion. So we are in a well-mixed environment with minimal medium lacking adenine or lysine so that the two strains are forced to cooperate. We allow them to grow up. Presumably, cheater mutants will arise. And because I've shown you that cheaters have a fitness advantage over cooperators, you would imagine that the frequency of cheaters will rise over time. And eventually, the entire community will be crashed. So, but is that what we observe? So now I'm showing you number of community doublings as a function of time. So if community is growing exponentially, the number of doublings will increase linearly with respect to time. So what we saw is a quite heterogeneous outcome. So all these communities start from the same master mix. And orange-colored communities grow fast, while gray-coded communities grow slowly. And because we can use flow cytometer to distinguish these three strains, we analyze the outcome at the final time point. We found that fast-growing communities were dominated by cooperators, while slow-growing communities were dominated by cheaters. We then performed the whole genome sequencing, and the picture became very clear. So what happens was that cooperators and cheaters, they're competing for the very limited lysine from the partner. So they were both sampling from the same pool of adaptive mutations. So if cheaters by chance sampled a better mutation than the cooperator, then cheaters will dominate and the communities will be crashed, it will stop growing. On the other hand, if cooperators happened to sample a better mutation than cheaters, then cooperators would dominate. And these would eventually even purge cheaters. So these communities grew as if there were no cheaters. And we term this adaptive race. Adaptation to stress can lead to stochastic cheater purging. This has been observed in natural microbes. For example, Pseudomonas fluorescens under iron limitation would make siderophores, and these are high affinity chelators for irons. But both cooperators and cheaters would compete for siderophore iron complex. And because cheaters have a huge fitness advantage over cooperators, we can consider if cheaters start at 1% frequency, because of this huge fitness advantage, they will increase in frequency. However, if we add phages, so note phages have nothing to do with this cooperation through siderophores. So if we add phages, then cooperators and cheaters both need to come up with mutations that would protect them against phages. And because the starting population of cooperators is far more than cheaters, it's much more likely for cooperators to sample a better mutation, better phage-resistant mutation compared to cheaters. And that would allow cooperators to drive cheater frequency down. So I've just shown you an example where by getting rid of all known pro-cooperation mechanisms, we were able to discover adaptive race as a new mechanism that protects cooperators against cheaters. So now I would like to switch gear to talk about the importance of quantitative thinking. And we learned all these lessons from our synthetic communities. An outstanding question in biology is, how might community level properties emerge from cell-cell interactions? So let's look at the Cosmo. So I'm plotting cell number against time. And the red-green indicates the dynamics of the two cooperators, and the black is the dynamics of the total population. If we observe this, we see that initially there's a lag phase, and then the both strains and the total community grows exponentially. And we call the slope the steady state growth rate of the community. So after some mathematical calculations, we found that the steady state community growth rate is the square root of lysine release rate per cell times adenine release rate per cell divided by adenine consumption per new birth per cell and divided by lysine consumption per cell. So community growth rate is determined by metabolite release and consumption. So this actually provides an example of how quantitative thinking can inform us what to measure. Note that in this example, we do not have phenotypes such as the affinity of the two strains for adenine or lysine. And the reason is actually intuitive once we think about it, right? Because in this community, the cells are limited for the metabolites provided by the partner. And if the partner releases metabolites slowly, the affinity, right? The affinity for metabolites, that is how fast the cells can take up metabolites, is actually irrelevant, because the rate limiting step is how fast the partner releases metabolite. So in other words, if you're waiting for your partner to give you meals, right? Eating meals faster does not influence how fast you can grow. It's how fast your partner can release. So we measured phenotypes from batch cultures. So the way we did it was that to measure consumption, we just give them metabolites and see how many cells can be produced. For release, we just use starved cultures. And note we have to measure these phenotypes in monocultures, because in co-cultures, the metabolites, the amount of metabolites are so little that they're not detectable. So then if we measure them in monocultures, we decided to measure them under starvation conditions, because otherwise it will be, because the communities grow kind of slowly, very slowly, double every 6 to 8 hours. So we thought that starvation cultures would be a good approximation of very slow growth. So when we measured those phenotypes and put them into that formula, the prediction is much faster than the experiments. You might think that, well, the difference is not actually that large, but I want to remind you that growth is exponential. So even small difference in growth rate will amplify into exponential divergence between predicted dynamics versus experimental dynamics. So that was kind of discouraging, right? This is a very simplified system, and we could not quantitatively understand how fast community grows. But we thought there could be many reasons why this discrepancy exists, so we kind of actually gave up. But at the same time, we were baffled by some other observations, right? So we were actually doing a very standard assay, trying to compare mutants with ancestor. We want to measure the release rate per consumption. If a mutant can release more per consumption, then this mutant is more helpful to partner than the ancestor, right? Because per consumption from partner, it actually gives partner more metabolites. But the problem is that using those batch culture assays, we keep getting very noisy experimental data. So we have huge error bars that prevented us from comparing ancestor with the mutant. So this actually troubled us for over a year. Eventually, we realized that perhaps if we use batch cultures to measure these phenotypes, and the phenotypes from these batch culture measurements could not explain experimentally observed cosmal growth rate, then perhaps the batch culture assays are not good. So we spent another year building chemostats. So chemostats are continuous culture devices that would allow us to control the growth rate of these cells. Right? So we can control them to grow double every 6 hours, for example, to mimic the community environment. And then once we did that, the model prediction experiments matched beautifully. And thus, phenotypes need to be measured in continuous cultures that mimic the community environment to achieve quantitative understanding of community level phenomena. But once we use the chemostat assays, the results become much more reproducible. So we indeed find a mutant that releases more per consumption than ancestor. And thus we found a mutation that can help partner to grow better. So I hope this vignette illustrates that mismatch between model and experiments can alert us that we're missing something very important. In this case, is that our batch culture assays are not good enough. And this in turn rescued another project. So I want to thank you very much for your attention.
Good morning, everyone. I'm Dr. Ryan McClure, a microbiologist at the Pacific Northwest National Laboratory. And it's my pleasure this morning to introduce the next speaker in our series. Dr. James Carruthers attended Yale University and received his PhD from Harvard before doing postdoctoral work with J.D. Kiesling at UC Berkeley and the DOE Joint Bioenergy Institute. Currently, he is Dan Evans Career Development Associate Professor of Chemical Engineering, an Adjunct Associate Professor of Bioengineering, a member of the Molecular Engineering and Sciences Institute, and the co-director of the Center for Synthetic Biology. His current work integrates computational modeling, CRISPR-Cas network engineering, and RNA aptamer biosensor design for applications in synthetic biology. The broad goal of his research group is to develop genetic control systems and to engineer microbes and cell-free systems to provide new sources of industrial and medically important chemicals and materials. Today, he's going to be discussing his work using CRISPR programs targeting bacterial metabolism.
Hello, my name is James Carruthers, coming to you from the University of Washington. Today, I'm going to tell you about some of the work we've been doing in our group to develop multi-gene CRISPR activation and inhibition programs to engineer bacterial metabolism. In our group at the University of Washington, we essentially do three things. We use large-scale computational simulations to design metabolic control systems. We build CRISPR-Cas transcriptional programs to implement those designs, and we build programmable aptamer biosensors as genetic controllers and as technologies for reading out those designs. All this work is aimed at engineering bacterial and cell-free metabolisms to produce medically and industrially important chemicals. The development of CRISPR tools for rapidly programming gene expression promises to accelerate the development of bio-based production systems. The application of CRISPR tools for transcriptional repression, that is CRISPR inhibition in bacterial metabolic engineering, is well established. By comparison, the creation of CRISPR tools for transcriptional activation in bacteria have really lagged. So we've been working now for a number of years with our collaborator, Jesse Zalatan, here at the University of Washington, to develop new tools for effective bacterial CRISPR activation. And we've done this by identifying effective transcriptional activators, by systematically defining rules for effective bacterial CRISPR activation, and by creating approaches for encoding functions in engineered guide RNAs. And so we've shown that these CRISPR activation tools can drive expression of multi-gene biosynthetic pathways from multiple promoters and in multiple hosts, including E. coli and Pseudomonas putida. In my presentation to you today, I'm going to outline some of our most recent results and highlight some strategies for integrating CRISPR activation and CRISPR inhibition to create new capabilities for engineering pathways in bacterial systems. Now, in principle, there is a vast space of medically and industrially important chemicals and materials that can be produced using biology. And in fact, it's not always appreciated that bio-based chemical production is already a $160 billion industry in the United States, and that we're on track for something like 25% of all chemical production to be bio-based by 2025. Now, of course, there are a number of notable recent successes in applying synthetic biology and metabolic engineering for applications in bioproduction. But the fact is that these remain challenging, time and resource intensive undertakings. And at a really basic level, we can understand this as being, the fundamental challenge here is that you have to program the activities of large numbers of gene products, and they have to all function together as a system inside a cell. So this really is a systems level engineering problem with all that entails. Now, there are lots of different ways to kind of think about how or why this is hard. I'm going to frame the problem in the following way. When we're going to introduce new multi-gene functions into a cell, we're doing this in the context of an existing really complicated network, right? So the underlying, of course, there's a genome, and the genome is really a network of networks where the cellular function is determined by the genomic links and the regulation. And so if we just introduce new functions into a network that looks like this one, in general, we're going to cause perturbations to the network. And a lot of those perturbations will be unpredictable. And so when we're doing this kind of process of introducing new metabolic functions to an existing network like this, understanding or being able to understand what the combinatorial design space is that's both accessible to us and then that we actually want the particular manipulations that we want to make in order to harness metabolism to produce these chemicals and materials, that's a really challenging undertaking. Now, I want to drill down just a little bit and make give you a more tangible example or illustration of why this is hard. So in my own lab, we've organized a lot of our efforts around the production of industrial aromatics, including molecules like this one, p-aminostyrene. It's a vinyl aromatic whose polymers have tunable optical and mechanical properties that are useful for a lot of different industrial applications. Now, in principle, we could make p-aminostyrene from glucose in about 15 steps if we introduce about 5 or 6 non-native or heterologous gene products to a metabolism like E. coli. In practice, though, when we introduce these new functions, we create a series of control problems. So there are intermediates here like p-aminophenylalanine, so PAF, that's readily diverted from the cell. There are other intermediates that are toxic to the cell at relatively low concentrations, like p-aminocinamic acid. If you overexpress some of the enzymes in the pathway, that has a deleterious effect on cellular metabolism, and so on and so forth. I'm giving you these examples in this particular pathway, but these are the kind of general class of control problems that you'll encounter anytime you want to engineer metabolism to produce something that the cell doesn't produce natively. Now, our strategy, or our goal really, is to create designable expression tools that we can use to rapidly program the timing and expression levels of large numbers of genes in these systems. So we want to use these to identify, build genetic programs that maximize the amount of flux into our engineered pathways, then program variations within the pathways themselves so that we maximize the conversion of the substrate into the final product, and at the same time turn down expression of genes that would otherwise divert either intermediates or products from the pathways that we care about. So our general approach is to use nuclease defective Cas9, so dCas9, with a guide RNA that specifies a target site on DNA. So if we target this complex to a promoter or an open reading frame, This will generate CRISPR gene repression, so CRISPR inhibition. To enable simultaneous activation, we use modified guide RNAs. We call these scaffold RNAs or scRNAs that have a 3' hairpin that recruits a transcriptional activator that's been fused to an RNA binding protein. And so we can express multiple guide RNAs and multiple scRNAs to inhibit and activate genes at the same time. So again, sgRNAs or small guide RNAs targeted to a promoter or open reading frame will result in CRISPR inhibition. And scRNAs targeted to an appropriate site upstream of a promoter will generate CRISPR activation. So now that I've kind of given you a general outline of the kind of big picture problem, why it is that we want to develop these tools for building multi-gene programs, in the next part I want to show you how we've developed some new rules for generalizing gene regulation in bacteria. We started this project several years ago by screening, and what we did is we screened candidate transcriptional activators. And by doing that, we were able to develop a new system for CRISPR activation-directed transcriptional activation in bacteria. And so this system really gave us the basis for building fully programmable platforms to target some genes for activation, again, and some genes for inhibition. Now, what I'm going to begin with a few more results from kind of drilling down in some more detail and some more recent work. And I want to begin with some of our results investigating questions that came out of that initial description of the system. And here are a couple of them. Which promoters can we actually activate using CRISPR activation in bacteria? What's the impact of the promoter sequence or the presence of transcriptional repressors? Can we gain a clear idea of where exactly we have to put the CRISPR activation complex relative to the transcription start site to get effective transcription initiation? Our hope was that we could learn to kind of effectively activate genes from synthetic promoters, that is, engineered promoters that were controlling our heterologous gene products, and also develop a set of rules that would allow us to activate genes from within bacterial genomes. So the first question that we asked with our new system was whether the intrinsic promoter strength affects the level of CRISPR activation that can be achieved. And so for these experiments, we used a test set of minimal promoters that spanned a 200-fold range in basal expression levels, from barely detectable to kind of maximal CRISPR activation, which incidentally is comparable to strong tetracycline-induced expression, if that means anything to you. And what we found is that CRISPR activation is sensitive to promoter strength. Perhaps, not surprisingly, you can't significantly activate genes with high levels of basal expression. And at the other end of the spectrum, we weren't able to significantly activate expression from promoters with the weakest levels of basal expression either. But then there's kind of this Goldilocks range of not too high that it can't be activated and not too weak that it can't be activated. And that's our kind of working expression space for using these tools. So in our original paper describing the CRISPR-A system, we'd seen a really strong dependence on the distance between the transcription start site and the guide RNA target site within the promoter region. And so now what we're doing is adding more resolution to that analysis. And we found something interesting. That is, there's kind of a well-ordered rise, then fall, then rise again in activation as we move away from the transcription start site, which is shown at the right side of this figure. So we're moving away, and the numbers indicate kind of distance from the TSS. And we'd seen previously that -81 was kind of the optimal distance, but now we can see that there's quite a bit of activity that can be recovered at -91, a little bit if you move closer at -70, and even a little bit at -102. Eventually, it seems to fall off completely. And what we now know, or how we interpret this, is that we think that the CRISPR-A complex is rotating around the DNA, and that it needs to be on the same side as the polymerase, which gives rise to these peaks of activity. Now, through these experiments and a number of others that I don't have time to present today, a picture of the complexity of the task of activating endogenous promoters emerged. But we reasoned that it should be possible to apply what we'd learned to identify some targets in the E. coli genome that could plausibly be activated using CRISPR activation. So for this experiment, we selected a subset of genes with the right protospacer adjacent motif kind of target sites for S. pyrogenese dCas9 and an expanded PAM recognition dCas9 variant. We wanted basal expression levels that weren't too low, not too high, and that were regulated by sigma 70, which isn't much of a constraint in E. coli. And then we measured CRISPR activation from endogenous promoters. And what we found is that our predictive rules enabled activation from 4 of 6 endogenous E. coli promoters that we tested. And so, there's still more we'd like to understand about the rules for activating endogenous genes, particularly, whether we can make CRISPR activation or CRISPR-A directed gene expression levels quantitatively predictable. Even at this stage, we estimate that something like 80% of the E. coli genome may be targetable using these tools and approaches. So we think we're on the right track. All right, so now we've got in hand some new rules for improving and generalizing synthetic gene regulation. So now we're going to turn to the question of whether we can use these rules to create a set of new tools for building multi-gene expression programs. So, we've seen that bacterial CRISPR activation is sensitive to a number of factors. We wanted to know whether we could incorporate these design rules with some additional constraints to create synthetic promoters that could be targeted for activation through the regulated expression of a synthetic or engineered scRNA. So namely, we have a set of design rules for we want to build these promoters so that they are not cross-activated by one of the other scRNAs. We want to make sure that they don't have any binding sites for known transcription factors and that they don't have any kind of homology to one another to enhance genetic stability. So our initial set here contained 4 engineered or synthetic orthogonal promoters, and we got as much as 50-fold activation when we delivered the scRNA to the cell. So this is a great sign, but if you get a look across at the quantitative levels of CRISPR activation in the set, we wondered why the other promoters in the set were not able to achieve all the same levels of activation as the highest one, J3. So we had seen in some of our other work that co-transcriptional folding trajectories can have a significant impact on RNA functions, and that a lot of these impacts result from what are best understood as kinetic effects that are governed by the sizes of the energy barriers that separate the structure that an RNA folds into from at the end of transcription from the structure that's needed for function. And so we wondered whether differences in the spacer sequences, that is the sequences that actually target the scRNA to a particular place in DNA, were impacting the folding of the scRNAs, and whether these differences could be responsible for the effects that we see. And so we tested this by building a set of 30 different promoter scRNA pairs. And in fact, we found that the effectiveness of the scRNA-directed CRISPR activation depends in a significant way on the predicted kinetics of scRNA folding. That this term is it goes a long way toward predicting whether an scRNA is going to be effective for generating CRISPR activation. Okay, so now if we go back and we use our now new sequence and structure-based design rules, we now see that we're able to expand this set. And so now we've got a set, now I'm showing you 2 additional synthetic promoters that are now achieving the same level of expression as J3, which was previously the highest one. And in fact, we've now used this approach to generate a set of 8 different orthogonal CRISPR-A promoters that can be targeted independently for programmable expression. Now, there's one last capability that we wanted before we move on to some applications, and that is we want to be able to tune the level of expression without modifying the promoter. And we want to do this by just changing the expression programs, but through the delivery of different guide RNAs. And so our idea here was that truncating the 5' end of the spacer sequence, so that is the RNA sequence in the scRNA that targets the Cas complex to the promoter, could reduce the affinity of the scRNA for the engineered promoter, which would reduce the level of transcriptional activation. And so we applied these kind of sequence and structure-based design rules to create this set of truncated scRNAs. And now we're able to generate tunable levels of CRISPR activation. So we can go from 50-fold activation on the high end, and then medium and low, and kind of across the set. So now we've got the ability to target orthogonal CRISPR-A promoters independently that are programmable and tunable. Okay, so what can we do with them? So here's a test. We're going to use these to program combinatorial variations in gene expression. So now we're putting the expression of RFP, BFP, and GFP under the control of one of our engineered promoters. And then we're transforming cells with a combinatorial library of scRNA programs that have scRNAs that are going to generate high, medium, low, or off-target or no expression at all three of the engineered promoters. And as expected, and kind of gratifyingly, we see that only basal expression in the off-off-off condition, but then predictable variations in reported gene expression that depend on the identity of the scRNA program being delivered to the strain. And that this really shows that the expression of multiple genes can be independently tuned using these multi-guide CRISPR-A programs. Okay, so very briefly, now I want to show you how we can use these tools for applications in bioproduction. And so in this experiment, we're using CRISPR activation to independently regulate pathway gene expression in a pathway that's engineered to produce lacto-N-tetrose, or LNT, which is one of the most abundant human milk oligosaccharides and a target for the creation of new infant formulas. And it's expensive to synthesize at scale. So now what we're doing here is driving the expression of 3 heterologous genes expressed under the control of the same promoters I showed you in the previous slide. And now what happens is, you might think of this as a kind of focused combinatorial library, so a subset of all the possible scRNA library designs of a kind of high, medium, and low that would be possible. And what we see is that when we induce expression of the scRNAs, we're able to generate significant amounts of LNT, something like 160 milligrams per liter. And I'd also make the point that, interestingly, significant LNT production is only observed with the low, high scRNA program, which illustrates an important point and that's consistent with lots of observations in the field, that generally speaking, the highest levels of production from an engineered pathway are rarely obtained from the highest levels of gene expression. We really do have to balance the supplies and demands for cellular resources. And, of course, this also shows that we can use our tools to program gene expression in multi-gene pathways that are engineered to produce target molecules. So it gives us a new ability to program those combinatorial variations that and gain control over the timing and expression of multiple genes. All right, so in the last minute or two, I want to show you that we're able now to use these tools to develop non-model microbes as bioproduction hosts. Now, there are a number of non-model bacteria that have proven useful for bioproduction applications, and there are a number of efforts underway to develop new microbial hosts that have desirable either metabolic capabilities or that can utilize different target feedstocks that can withstand harsh bioprocessing conditions. And interestingly, the RPOA RNA polymerase gene product that is the target for interaction with our modified SOX-S transcriptional activator is well conserved across bacterial species. And so this really give us the idea that it should be possible to port these tools, CRISPR activation tools from E. coli, into diverse bacterial species. Why would you want to do that? As I said, a lot of these microbes have different abilities to withstand both substrates and products that might be the targets of metabolic engineering. In fact, in this pathway that we're engineering to produce p-aminostyrene, the soil microbe Pseudomonas putida is something like 3 times more resistant to the presence of one of these pathway intermediates, p-aminocinamic acid, than E. coli is. And so this, we think, means that it's going to be a good chassis or a good starting point for engineering a platform to produce this molecule. So what we've done is implemented CRISPR activation in Pseudomonas putida using the same system that we previously developed in E. coli, by developing a methodology to transfer CRISPR-A to a new host. So we optimize expression for the CRISPR-A components and then apply these rules for effective CRISPR-A based on systematic characterization. And so we see that not only can we build these new programs, but also that the levels of pathway gene expression are suitable for metabolic engineering, and that these can be readily achieved using our new tools. So again, this is another one of those, we think we're on the right track for being able to use these tools to develop new non-model microbe hosts. All right, so to briefly summarize what I've shown you today, we've seen that the requirements for bacterial CRISPR activation target sites do pose challenges, but our data also demonstrate that CRISPR-A has the potential to be effective at a broad range of target genes. So by systematically defining the rules for effective CRISPR-A sites, we've identified new strategies for improving and generalizing synthetic gene regulation in bacterial systems. We now have a set of tools and emerging capabilities for engineering multi-gene CRISPR-AI programs in bacteria. These CRISPR activation inhibition programs can be coded as genetically compact sets of scRNAs and sgRNAs. And the CRISPR-AI programs themselves can be used to specify a wide variety of functions. We can activate non-native genes, we can activate and repress endogenous genes, we can implement dynamic expression programs. Of course, we're still learning about these systems all the time, and so I don't want to give you the impression that we've solved all the challenges in CRISPR-AI program engineering, but these tools really are giving us new opportunities for developing bacteria as platforms for bioproduction. Now, ultimately, what we want to do is combine these tools with multi-omics data collection and machine learning, and then that would give us kind of the ability to fully develop multi-gene CRISPR-AI programs that could dramatically improve the ability to engineer bacteria for bioproduction through accelerated kind of design, build, test, learn cycles. So I will stop there and thank everyone for inviting me to give a talk and participate in this session, and I look forward to your questions and the further discussion.
Professor Caroline Aho Franklin earned a BS in chemistry from Emory University in Atlanta, Georgia, and a PhD in chemistry from Stanford University. She then did postdoctoral work with Professor Pam Silver in the Department of Systems Biology at Harvard Medical School. Until 2019, she was a staff scientist at Lawrence Berkeley National Laboratory in Berkeley, California, and has since joined the faculty of Rice University in Houston, Texas, as a professor of biosciences with a joint appointment in bioengineering. Professor Aho Franklin was named as a recipient of the Women at the Lab Award in 2018 and as Cancer Prevention and Research Institute of Texas Scholar in 2019. Her interdisciplinary and collaborative research program focuses on exploring the interface between living organisms and non-living materials and engineering this interface for applications in energy, environment, and biomedicine. Today, she'll be discussing her work on bacteria and macroscopic materials.
Hello, everyone. Today, I want to share some very recent work from my research group in which we've used synthetic biology to program bacteria to grow into macroscopic materials. And why I'm so excited about sharing this work with you is not only because these are engineered living materials are multifunctional and macroscopic, but in making them, we believe we've identified design rules that we hope will be generalizable and allow us to program many different types of bacteria into materials. So one of the things, our overarching goal is to create biomaterials with tailorable function. And this is the emerging area within synthetic biology called engineered living materials. And the idea is shown very nicely here on this slide, where just like in nature, seeds will actually grow into, for example, a tree, which has structural and mechanical properties and is macroscopic, but all of this synthesis requires very low energy inputs, just water and sunlight and a few nutrients. We, in the area of engineered living materials, seek to copy this, seek to create a single progenitor cell, an engineered cell that can grow and self-assemble into a structure that has mechanical, structural, and physical features that we have dictated. And we hope that this is a macroscopic material. So that's our overarching goal. We've actually made some significant progress towards that, especially in the area of assembling, and there's been some recent work in the area of macroscale engineered living materials. Now, it's much easier, since we're much better at engineering bacteria, making small microscopic structures has been pretty straightforward. But there's been some great work on engineering macroscale structures. For example, Neil Joshi and colleagues have been able to make aquaplastics from curly fibers out of E. coli. Chris Voigt and coworkers have made printed materials based on spore-forming bacteria that encapsulate spore-forming bacteria. Tom Ellis and Tim Liu and their co-workers have made a series of materials composed mainly of bacterial cellulose, but that also include living yeast cells and can actually function as sensors. So one of the things, we've been really inspired by the diversity of different functions and types of materials that we've made, but one of the things that has been lacking is the ability to make these really from the bottom up. In all cases, we've actually needed to either introduce chemicals or do some substantial processing to make these materials, as opposed to the seed that just grows with minimal input. Or conversely, in the case of the cellulose yeast materials, because it's built around a material that naturally forms, we haven't been able to tune it and to really control the structure in the way that we would really envision from a bottom up as opposed to a top down approach. So we really wanted to make a bottom up approach for assembling these macroscopic materials. And our approach was to figure out how to encode interactions between cells, weak interactions at a high density, so that they could work, so that these interactions could cause the cells to slowly form a large macroscopic material. So the foundation for our approach is actually the surface layer or S layer of the oligotrophic bacteria, Colobacter crescensis. And one of the really, while there are many cool things about Colobacter, one of the coolest is its S layer. This is a crystalline lattice that is found on the outermost surface of the cell. And if you look by EM, you can actually see this hexameric 2D crystal that's composed of proteins. And this S layer is basically composed of 3 domains: an anchoring domain that anchors it into the peptidoglycan, a domain that forms these beautiful hexamers called the crystallization domain, and a domain that's actually responsible for secreting the protein outside the cell. And I just want to give a little plug out for M Systems. Our work in actually using the secretion part of RSAA to make an extracellular matrix protein was published by M Systems, and that was a very positive experience, and I would be happy to chat with anyone about it. Okay. So our approach was to take the S layer, and because it provides such a high density display approach, and introduce self-interactions by replacing the crystallization domain. So we kept the N-terminal domain right here, and we kept the C-terminal domain. The C-terminal domain also introduces weak interactions between the cells. And then we introduced a long flexible linker that would move these interactions from close to the cell surface to further out, so that we could improve the accessibility of those interactions. And we used an elastin-like polypeptide that has the sequence repeated 60 times. And lastly, we introduced a functionalization tag, the spike tag, so that it could be functionalized with any spy catcher containing proteins. So that was our design, and we simply used genomic editing to change the locus, the native RSAA locus in the Colobacter genome. And when we did that, we found that we were able to make bottom-up de novo designed ELMs, which we call BUD ELMs. And these were centimeter scale materials that formed during the growth process. So here's we're looking at the bottom of a flask after about 36 hours of growth. And in the wild type strain, you just see a planktonic culture, a dense planktonic culture. But when you've now changed the single domain and introduced the ELP and the SPI tag, what you see are these small materials that are about 0.5 centimeter to 1 centimeter in diameter, that are freely floating in the media. These, we can show that these new bud materials actually form from the protein, and that the protein underlining their formation is actually on the cell surface. So what we can do is, using the SPI tag interaction, is incubate these cells, engineered cells, or the wild type with GFP SPI catcher. And what you see is, in the case of our engineered strain, the GFP binds to the outside surface of the cell, lighting up the extracellular surface, whereas in the wild type, we don't see any significant staining. And here, shown in yellow, is just the presence of the cell body through MK fluorescence expression. So the other thing we can see is not only is to actually use atomic force microscopy to actually dive in and take a closer look at the cell surface. And what we can find is that when we look at the wild type bacteria, we see this beautiful hexagonal lattice that is our S layer. If we've deleted it, we see a slightly rougher surface, but with our bud ELM surface strain, we see a substantially rougher surface that has projections that are much higher into a solution. And that's reflects this projection from the ELP on the engineered bud protein. Okay. So if we dive in at looking not just at the individual cells, we can actually look at the entire material and show that it has a hierarchical structure. So what Sarah Molinari, a very talented postdoc in my group, did is she actually gently opened up these materials and again stained them with SPI catcher GFP. And what you see on essentially the, now this is sort of hundreds of millimeters, you actually can see that the GFP and the cells are stained very somewhat uniformly throughout the material. So you see both channels at this scale. However, if we zoom in to this little fragment, what you'll see is there are actually regions of cell density, here shown in yellow, that are surround these matrix regions, is what we call them, that are of stain with our SPI catcher GFP. And so we believe our bud protein. Okay, and you see that that region is actually also absent. There are not cells in this region. And again, if you zoom in at sort of the single cell level, once again, you see that bud protein on the surface of the cells, as we saw in the individual bacteria. So what this, to our surprise, we got not only the structure that we intended, this attachment between cells, but we got this matrix, which seems to be important in wrapping the material together into a much larger macroscopic material. So this extracellular matrix is predominantly composed of protein. I showed you that it stains with that SPI catcher GFP. And here you can see that if we stain with a Congo red stain that predominantly stains amyloid protein fibers, we see that we end up with staining in those matrix regions. Whereas if we stain with molecules that are either for lipids or for carbohydrates, we don't see that same pattern of staining. And indeed, instead we see just random staining that doesn't overlap well with our matrix areas. So that shows us this matrix is indeed composed of protein. So the next thing I want to share with you is our discoveries about how these actually form. And this is the work of Bobby Tesorio, a very talented graduate student in my group. And what he discovered by actually watching and taking photographs of these of the bud ELM strain during growth is that at first they form just a planktonic culture that lasts about 12 hours. And then around 12 to 16 hours, they form this pellicle that floats at the air-water interface. That increases in thickness and density, and actually then eventually collapses on itself and sinks to the bottom of the culture as our final material. So he also realized, having watched this process, that he could modify the morphology of this of our bud ELM by changing the shaking speed, and that's wrapped up here in this modified volumetric power parameter. And what I'm showing you is that as we increase this volumetric power, we go from having small diffuse material to large pieces of material, a single large piece to multiple small pieces, and then again, small fragments. So there's a sweet spot in terms of material formation in these ranges of volumetric power. So we also really wanted to see what these bud ELMs can do in terms of acting as a living and a material. So one of the things that we did is that we actually dried this material out. And then after drying it out on the bench, put a small piece back in culture and tested whether it could indeed reseed its growth, just like the seed of a tree. So what we found is that if we left it out for 7 or 14 days, we could easily form 100. We found that 100% of the times we tried, we got material. But after 21 days of sitting out of culture, we found that only about 33% of the time did we get this bud ELM material to reform. So that tells us we do have these seeds. We can reseed its growth. The other thing we did is collected material from multiple flasks and then showed that we could actually combine it and extrude it into different shapes and also mix it with inorganic materials. Here I'm showing you with glass beads into now solid organic, inorganic composites. So it can be reshaped. So the last thing I want to kind of share with you about these that we've discovered with these bud ELMs is that we can really change their morphology, not just with shaking conditions, but with growth conditions more broadly. So you can see that here are some bud ELMs that are grown with sucrose or glucose, but we get this really interesting structure when we grow them in xylose. And when we pull it out of the flask, it looks like a twisted rope that is now multiple centimeters in length. And this is an elastic rope, so you can stretch it. And that arises almost certainly from the elastin-like protein. So we feel like we are just at the tip of the iceberg to learn all the different things that we can do with this material. So to summarize, we found that we could use a high density of weak interactions to encode bottom-up formation of these living materials. That these living materials are hierarchically structured and multifunctional. And I think what's really exciting here is that we have a technological platform as well as design rules for making macroscopic materials from the bottom up. So we're looking forward to these ELMs proliferating and generating new shapes and functions. So with that, I want to just thank the many people that have been involved in this work. So this work built upon a DARPA engineered living materials project in which we tightly collaborated with Paul Ashby and Kathleen Ryan and had major contributions from Mary Michael Charrier, Maria Orozco Hidalgo, Dong Li, and Nicholas Che Hayano. And the work that I showed today has been mainly carried out by my group, Dr. Sara Molinari, Bobby Tesorio, and Jayatri Saman. So with that, I will thank you and look forward to talking to you and answering any questions you have.
Okay, well, while we're all just kind of digesting those three talks and kind of turning our cameras on and getting ready for our Q&A panel, I just want to thank all three of the speakers again. Those were really great talks, and we learned a lot of really interesting stuff. So we really appreciate you sharing your research with us. And I see that the Q&A panel, the chat feature is pretty active. Folks are posting questions and our speakers are answering them. We also had some other questions that showed up in the other chat. And so I'm going to leave the Q&A ones for the moment, and we can answer those by text kind of as this is going on. I'll just kind of focus on some of the other ones that were posted in the chat. So I'm just going to kind of read the questions that I've been getting, and our three speakers can jump in with their great answers. So Wenying, one of the questions that came up that I don't think was in the text chat there, which is, how does this use of continuous culture versus batch effects, where you saw that when you switched to continuous culture, you got results that I think agreed much more readily with your models? How does that kind of port to understanding how these kind of interactions might take place in like a natural system, which aren't really fully batch or really fully continuous culture? Or what other steps are you kind of taking to sort of apply or port what you've learned in these laboratory systems to how this might be happening in a natural system of a cheater and a cooperator?
Yes, that's an excellent question. Can you hear me first of all?
Yep, I can hear you fine.
Okay, good. So the difference between batch and the continuous culture. So batch culture, you have only two types of batch cultures. You either give them tons of metabolites, so they're growing at the maximum growth rate, or you give them no metabolites. That's the only way you can keep the environment constant. But for continuous culture, because chemostats is constructed very cleverly, so you can control, for example, the growth rate and the density, so that really allows you to tune your environment to be similar to the community environment. So the big lesson we learned here is that the cell phenotypes, the physiological phenotypes are very sensitive to the environment. And to achieve quantitative understanding, right? I mean, for qualitative understanding, maybe you don't need to go to such great length. But if you want to understand a phenomenon quantitatively, it may be necessary to recreate the environment that they are, that the community-like environment, to measure the phenotypes, so that you can try to understand how phenotypes of the individual species in monoculture situation would culminate into the community level property. So, and whether so my feeling about the complex natural communities, I mean, the environment would be far more complex, right? Because mostly they'll be spatially structured, and the environments will be very different from each other. So I would argue that for complex communities, I don't think the time is ready for quantitative understanding. But still, like mathematical modeling can be used for qualitative statements. For example, what encourages species coexistence? For example, certain, like how does a resource the richness of resource correspond to species diversity? There are questions like this, right? So you can get a qualitative understanding of what's going on. So I still feel mathematical models can be very useful.
Yeah, definitely agree. So James, a question came in for you. So how accurate is or are the predicted kinetics for the small RNA folding? So how much folding space is there, and are there any ways to sort of improve these kind of prediction kinetics, or how do any of the maybe errors in the kinetics affect kind of what you're doing?
Yeah, no, that's a great question. So let me make sure I understand the question. I'll kind of repeat it back, but I think the question is, so in making these secondary structure predictions, and then in determining how long it's going to take to transition from one structure into the other, how much uncertainty is there in that prediction? And so I should say the kinds of calculations that we're doing. So these are really, we've actually tried a bunch of different kinds of calculations, and we examined a bunch of different parameters associated with guide RNA folding. And several of them kind of emerged as having the largest impacts on our ability to predict which guides were going to be good. And so what we're doing here is doing a barrier height calculation. So this is really how much energy do you have to, is there that separates the two states. So the state that the RNA folds into at the end of transcription from the secondary structure state that we know to be necessary for function. And so depending on the height of that barrier, so a bigger barrier means that this will happen less frequently. Now, so in doing those calculations, we found that we don't necessarily have to have a very high degree of accuracy, just because the scale of the difference is actually many kcals per mole. So we're talking about, the worst ones in our set might be 10 or even 15 kcals per mole worse than the best ones in the set. So you don't have to be very accurate for that. Now, that we have some other work I didn't talk about, but where we're using these barrier heights and kind of energy and kinetic calculations to design RNAs that have different folding rates, so that we can build kinetic control into biosensing functions and things like that. So in that case, the accuracy is kind of more important. But in this case, we get pretty good results, because really we tend to only want very good guides, or to know that we're not going to get bad guides. So that's a more binary kind of question. So the accuracy isn't quite as important for us.
Quite a wide target that you can hit. So
yeah, exactly.
Okay, I understand.
Exactly.
Thanks. Caroline, we had a question for you, which was, when you're looking at these species that you're sort of modifying to build these materials, there's genetic modification that's happening like at the DNA level. Have you looked at modifications at the other parts of moving from a gene all the way to this material? So targeting maybe the post-translational or post-transcriptional modifications, or changing the media types or the metabolite inputs, or anything like that?
Yeah.
How do you think, and maybe you haven't, but how do you think attacking those other parts might help improve things even further?
So this is some very fresh work, and so I really appreciate that question. And one of the things that we've really come to understand, and kind of even data since when I recorded that, is that in addition to design rules, we're coming up with assembly rules. So, for example, one of the things I talked about is how fast you're shaking. Okay, so one of the things that happens is this material starts to, I'll use the term that material scientists would use, it starts to nucleate and grow at the air-water interface. And then the shaking really controls whether you have in one big piece that kind of nucleates and grows and then sinks, or whether it have multiple pieces. And that's actually something called Oswald ripening. It's like whether, if you have oil in water, does it form one big droplet or lots of little droplets? And so the shaking affects how many little pieces we have. So we've also found that the air-water interface is absolutely essential. So if we use a detergent that disrupts the structure of the air-water interface, we lose our ability to make this material. So there's some rules we've been gleaning about assembly, that the air-water interface is really critical to initiate material formation, and that there are competing forces between creating the material and making it sink. So one of the reasons we think that we get that really twisted material is that it takes a real long time for that material to sink. And so it keeps getting twisted as it shakes. And that's why we get a rip. So we're really trying to learn not just, again, design at the genetic level, but assembly in the growth phase.
Okay, thanks. Wenying, one of the other questions we had for your work was these cooperation versus competition principles and phenotypes that you're looking at. How do those shift when you're looking at a well-mixed liquid culture, where there can be presumably interactions that span a wider range, or that look at involve a greater number of species, versus biofilms, where things might be a little bit more structured and a little bit more controlled. So, and does this influence the ability to predict the outcomes with your model simulations? Would we need multiple different model simulations for biofilms versus well-mixed cultures, or can we kind of port between the two?
Well, thanks for the question. It's a very good question. So I would argue, I would say that the outcome of a cooperator cheater in well-mixed environment is very different from that in a spatially structured environment. Because what happens is that in a spatially structured environment, where you repeatedly interact with your neighbors. So if your neighbors are cooperators and you're cooperators, right? So then you help each other out, you grow. But if you happen to land near a cheater, the cheater is not helping you, and then you die, and the cheaters die. So it's a repeated interactions. So in contrast to this repeated interactions in a spatially structured environment, in a well-mixed environment, regardless of how much you contribute, but everybody has equal access to the cooperative benefits. That's why cheaters are predicted to always win, because they have equal access to partner-provided benefits, yet cheaters grow faster than the cooperators. But we have shown that adaptation to stresses could sometimes allow cooperators to sample better mutations than cheaters and allow cooperators to purge cheaters. So in general, like the well-mixed environment is far easier to model because it's just ordinary differential equations. You can use MATLAB to solve it really quickly. But for spatially structured environment, if you want to understand the spatial organization of cells, it's much, much harder. So yes, of course, a partial differential equation is harder, but that's more than that, right? So if you want to model those cells, so remember, so if you're modeling spatial structure, you might want to model individual cells, how they arrange against each other. And because growth is exponential, you very quickly would be bogged down with tracking many, many cells, right, to the millions. And so actually for the spatially structured models, so we use parallel, we had to use parallel computing, and it actually takes a week to get to the pattern, and its experiments also take a week, so the simulation is a lot faster computation. But since then we have collaborated with some computational, some hardcore computer scientists from PNNL. So they could really do parallel computing in a more beautiful way than we have done. So they can cut down the simulation time from like 1 week to 1 day. It's still an outstanding question. Like when you deal with large systems, the computers do have trouble keeping up, just even tens of millions. So we stop at 10 million because after that the computers just become so slow. It's almost not worth it, right? But so that is actually an outstanding question. How would you deal with large systems, right? Where it's a very resource intensive to simulate.
So the modeling of a well-mixed system seems like it takes a lot less computer input than the modeling of a structured non-mixed system and just so I understood you, this model for the structured system, you're trying to predict the behavior of each individual cell?
It's more like how the cells arrange against each other. So like if I have red, green, and blue, initially randomly distributed. So you allow them to grow. So initially it's quite easy, right? It's like hundreds of cells. Maybe they will grow to millions of cells. And during that process, how would these the three types of cells arrange themselves, right, while they grow on piece of agar pad? So this is what I mean by spatially structured environment, because actually we're modeling 3D, the three-dimensional organization of the entire of the community. And for the well-mixed environment, it's just really straightforward ODEs, right? So yes, it's a computation a lot cheaper. Yes.
Yeah. I understand. Mm-hmm. Okay, great. Good to know. There's a lot of computational and lab work that are meeting up. That's great to hear. So James, when you were talking about some of the work that you were doing, especially with this milk protein towards the end, a lot of times if we're trying to make a particular molecule through biological means, at large scales, we're moving into large bioreactor vats, and this is a much greater volume, and it's carried out over a longer time frame. So in these kinds of systems, there's a lot of very specific, genetic bases for how these work. How can we constrain mutations and the organisms escaping out of the things that we want them to do to what might be best for them, especially when we have very large volumes and much longer time frames?
Yeah, sure. Well, I mean, that is certainly one of the biggest challenges in the field in general, in figuring out how you go from some some experiment, even from some strain that you streak out in a glycerol stock to, let's say, 10,000 liter tank or something like that. There are a lot of generations between there. I think in practice, the ideas of building systems that are and testing them for genetic resiliency to kind of environmental perturbations. Because the other thing that happens is that in the tank, the microbes experience really different environmental conditions. So, if you think of a giant tank, the experience of a cell at the bottom is going to be really different from the experience of a cell at the top, where pressure, availability of nutrients, all these kinds of things, even in a well-mixed tank, are just going to be very different. And so I think, these days, I think a lot of people are interested in identifying multi-gene phenotypes that can confer this kind of resiliency. I think that certainly we have the idea that building dynamic feedback control systems of the kind that are found in naturally occurring cells, so that our systems can also respond to changing demands and supplies for cellular resources, is going to be an important part of kind of making that next generation of bioproduction hosts more feasible than they have been in the past. But it's a terrific question. I think there are a lot of really interesting kind of science and engineering that has to happen before you can,
definitely
really do that. But yeah, thanks for the question.
Yeah, of course. So Caroline, when you're growing these communities and you're making these biomaterials, I was curious. So what happens if you go beyond 21 days? Do you keep getting the biomaterial, or do things start to, you're in a batch system, so do things start to crash out? At what point do we stop making the biomaterial and everything just starts to go into death phase?
Yeah, so I think I ran through a lot of material real fast. So what we did with that 21-day experiment is we took a piece of material out of culture and just left it on the bench for 21 days and said, hey, can we grow new material after it's been sitting around for 21 days? And
that's right. When you were growing, you said that was 36 hours?
So yeah, we were growing for 36 hours. So actually, so you're asking, sorry.
So what if we go past 36 hours? I'll rephrase the question.
So we actually have to, for kind of again, talking about assembly, what we see is there's also a relationship between the end morphology we get and how long we shake. So sometimes what we do is we get material, if we're shaking very relatively slowly, what we'll do is we'll get a material forming at the air-water interface. And what you have to imagine is kind of like a vortex, a whirlpool, kind of, because it's going around. If that whirlpool does not pull enough, that material just sits at the air-water interface. The boat never sinks. But if we shake really, really hard, what we do is fragment the material. So it's like fragmenting the boat, because the shear forces at the edge just start pulling it apart. And we also see that if the material is sitting at the air-water interface for very long periods of time, it just starts falling apart and crumbling into little bits that sink, but not the whole thing doesn't actually sink. So there's some really interesting aspects of how the basically hydrodynamic forces in the flask are shaping and essential for this material. So we're diving into some fluid mechanics that we have never dove into before, but it's fun.
And just in kind of thinking about the questions that were asked earlier, so this is in a batch culture. You said these are pictures of bottoms of flasks. So if you moved to a continuous culture where they always have fresh nutrients and they're always a little bit happier, do you think that you could continuously make this material, or do you think eventually you might bump into some issues?
I think so.
Or at least theoretically.
Honestly, we haven't seen, we need to do the experiment. You never quite know until you do the experiment. But we haven't, when we've gone out, when we're shaking very slowly and we don't form material, we actually don't see cheaters, for example, take over. Or so I actually kind of think that there's a shot. that we're going to do.
Great. Yeah, I like to hear that. I like to hear there's a shot. That means we can give you a moment and check out if it's working.
Yeah.
So Wen-Ying, one of the other questions we had was, so you showed at the beginning how you have cooperators and you have cheaters, and eventually, because these cheaters have this growth advantage, you switch over to a community that's almost all cheaters. But then you also kind of talked about some things that you discovered that can help prevent this, that the cooperators can do. So if you have a system that shifts over to mainly cheaters, is it ever possible for it to go back to mainly cooperators for some reasons, maybe some of the things that you showed here? And is there kind of this long-term cycle going back and forth that might be happening?
Yes, thanks for the question. So it's an interesting fact, because they are cooperators and cheaters competing, like sort of like sampling from the same pool of mutations. So it's actually the function of initial population size. So for example, if I start the community with the partner, we keep it fixed, just cooperator. But like this, the strain 1, we just keep it cooperator, and the strain 2, for example, 50-50 cooperator cheater. So that's 50-50 chance that cooperators will win or cheaters will win. But if for some communities where cheaters will take over, right? So now cheaters are like 1 to 10, but then cooperators still will have a chance of rising in frequency, but the chance is much smaller. It's a 1 to 10. So because it's actually really beautiful correspondence with the population size, because the larger population size you have, the more likely you could sample a better mutation. So occasionally we do see a cheater-ridden community will suddenly like take off. Or conversely, we also see communities where it's cooperator dominated, but suddenly cheaters take off. So it's actually very interesting evolutionary dynamics.
Great. So I think we have time for one more question before we go to the breakout rooms. And I kind of wanted to pitch this to both James and Caroline, which is the stuff you've shown has been, as far as I could tell, mostly limited to one species. What about introducing another species and spreading out those genetic modifications, or taking some of the work that Wen-Ying's doing and programming some interactions in a more stable community to kind of promote both of the goals of both your research? Do you think that's something that's possible, James?
Yeah, I'll make mine quick. It's something that we're thinking about a lot, and especially I didn't talk about this work, but we're interested in carbon efficient bioconversion, that is sequestering carbon dioxide and embedding that into products. And so, one idea is that you could spread the energetic burden around multiple cells, and maybe have a chance of doing this in a way that, even though proof of principle has been done in kind of a single organism, this is very inefficient from the standpoint of any individual cell, but maybe spreading the burden a little bit.
Yeah. So I really thanks for the great question. So one of the analogies I would make is that if you look at most naturally occurring biomaterials, they're formed from multicellular organisms, and different cell types have different functions to make that material. So I actually very much do agree that we should kind of look to nature and use sort of that multicellularity to kind of think about how we make our materials. So Caulobacter can grow in rainwater, it can grow in stream water, but it still needs a little bit of carbon. It would be great if we could give it CO2 and light, and we could get the job done.
Mm-hmm. Great. Well, again, thanks to the three speakers for answering these questions. This was a really good interaction. And I think if everybody looks in the chat, there's been something posted about talking to the speakers a little bit more, maybe if you had another question, or just kind of sort of virtually simulating the idea of walking up to them after the talk and just chatting with them. So there's a link there for these breakout rooms. A new one just appeared at the very bottom of the chat. So I encourage everybody to click on that and join one of these three rooms to talk to the speakers a bit more. Thanks, everybody, and thanks all the speakers. We really appreciate the work you're doing and sharing it with us.
Thanks for moderating.
Thank you.
Yeah.